Welcome back, friends. Here I am, Dr. Mlelwa, and today we are going to continue with our topic of gaseous exchange and respiration. In today's session, we are going to discuss about the electron transport chain. In the previous session, we discussed about the Krebs cycle, and we see how it is important, the substrates and the end products, all the mechanisms of the Krebs cycle. This is how we discussed it from our notes, also from the Campbell biology book. Now today we are going to discuss electron transport chain. It is called electron transport chain because the hydrogen carried by the hydrogen carrier from the Krebs cycle and from the link reaction, also from glycolysis, they are carried to the electron transport chain. Now how does this comes? It is because when the hydrogen carrier carries hydrogen, also it carries electrons. So at the starting of the electron transport chain, the hydrogen and electron tend to dissociate, and then the electron tend to pass through electron carrier, series of electron carriers, while the hydrogen is passing out of the series of electron carrier. At the end of the day, you are ending up with formation of water at the end of this chain. Now this is the summarization of how the process occur, and this diagram is taken from Understanding Biology book. So you can see what happens. So here you have NADH2, which is the hydrogen carrier. NADH2 reduce the NAD, and here is oxidized NAD. Now when NAD enters in the electron transport chain, NAD gets oxidized, so you get oxidized NAD, and then if it gets oxidized, it transfer its hydrogen to flavoprotein. So NAD transfer hydrogen to flavoprotein. It tends to reduce the flavoprotein by transferring its hydrogen to flavoprotein. And when hydrogen tends to transfer from NAD to flavoprotein, this leads to production of a single ATP. So this is the first ATP which is produced in the electron transport chain. From there, here again hydrogen hydrogen from a Krebs cycle, which is taken by FAD, enters at this level, not at this level. That's why FAD produces 2 ATP, because yenyewe inaingia at lower level. Unaona hapa NAD inaingilia hapa kwenye flavoprotein, lakini FAD inaingilia hapa. Now ninachokizungumza ni kwamba hydrogen ikishatoka kwenye flavoprotein hapa, ikishatoka kwenye flavoprotein inaenda kwenye nini? Kwenye coenzyme Q. Sasa inapokuwa inaenda kwenye coenzyme Q, hapa hakuna ATP inayokuwa released. Unaona? From there tukishatoka kwenye coenzyme Q, kinachofuata ni nini? Tukishatoka kwenye coenzyme Q, tunaenda kwenye cytochrome B. Sasa kwenye cytochrome B hapa, kabla hydrogen haijaenda kwenye cytochrome B, tuna separate. Now hii hydrogen ili iwe neutral lazima tu tuna proton pamoja na nini? Na electron. Proton maana yake ni positive charged. So that means tunakuwa pia tuna 2 electrons. So hapa kabla hatujafika kwenye cytochrome B, hapa kuna separation kati ya proton na nani? Na electrons. So the electrons, they are going to cytochrome B, and the protons zinatoka wapi? Nje. Kwa tunachokisema ni kwamba hapa kinachotokea kuanzia hapa kuja huku chini mpaka huku chini, kinachotokea ni electron transport chain. It's not hydrogen again. So mwanzoni at the start tuko nafanya transport ya vyote, lakini unaona kuanzia hapa kinachokuwa transferred mpaka huku chini, it's only electron. Huku chini electron again combine with a proton to form electron combine with proton to form neutral hydrogen, and that hydrogen combine with the oxygen to form water. So here at the end of this series of reaction, electron, proton and oxygen tend to combine to form water. So the reaction they are ending up with combination of electrons, protons and a half molecule for oxygen to form water. So this is the end of respiration. ATP are produced. As you can see here, we have 2 ATP. So NAD ataproduce 3 ATP, maana yake hapa ATP moja, ATP ya pili, ATP ya tatu. Lakini FAD kwa sababu anaingilia hapa, yeye ATP huku juu hamproduce, kwa ataproduce ATP huyu hapa mmoja na huyu hapa ATP ya pili. So from cytochrome B, unaweza kuona hapo diagram yetu hapo. From cytochrome B, kwa mfano hapo kwenye hii diagram yetu, ukianzia cytochrome B, then ukafuata cytochrome C, ukafuata nani? Ukafuata na huyu huku. Sasa kuanzia huyu cytochrome na huyu kuchini, hawa wote hawa tunaweza kuwaita jina la pamoja tunaweza kuwaita kama electron carriers. Kwa sababu gani? Wana carry electrons. Kwa wana carry electron kuanzia hapa hadi hapa. Kwa sababu hapa kulikuwa kuna separation, proton ndio ikapita huku, proton ikapita huku ikaja hivi. Lakini electron ndio ikafanyaje? Ndio ikawa inakuwa carried nao hapa. So unachokuja ukiona ni kwamba transfer ya electron kutoka kwenye cytochrome B kwenda kwenye cytochrome C, unapata a single ATP. Then transfer of electron kutoka kwenye cytochrome C kuja kwenye huyo hapa iron containing compound, unakuja kupata nini? Unakuja kupata ATP nyingine ya pili. Kwa unakuja kuona kwamba ndio maana tunaita electron transport chain. Kwa ni kweli at this piece kuanzia hapa kuja huku juu tuna transfer electron pamoja nani? Na proton. Lakini kuanzia hapa kushuka huku chini tuna transfer only nini? Only electrons. Ndio maana tunaita electron transport chain. So this explanation just summarizes what is the meaning of electron transport chain before we are going to discuss about every aspect of electron transport chain. So today we are going to discuss about the electron transport chain as a whole, and I know the session will be somewhat longer because of the process itself. It is very long and it is somehow complicated. So for the matter of understanding, and because I want you to understand what I teach, I will use a longer time, just about 1 hour, 1 hour and a half, so that you can understand what I am teaching or what I want to explain about the electron transport chain. Now, by definition, this is the series of hydrogen and electron carrier ending with oxygen. Series of hydrogen and electron carriers. Hydrogen and electron carriers ending with oxygen. Or in the means by which energy from Krebs cycle in the form of hydrogen atoms is converted to ATP. The means by which energy from Krebs cycle in the form of hydrogen atoms is converted to ATP. So how the process happens, the mechanism, and how the mitochondria perform this processing, we are going to continue. We are going to explain all of these in the next pages of our discussion. Now, this is our next page, which continue to explain about the electron transport chain. It's a mechanism or some of the few concepts which you must understand in the electron transport chain. Okay, there I just set the page well. Now, we are saying that the electron transport chain of molecule consists of flavoprotein derived from vitamin riboflavin. Now, you must understand this electron transport chain contain compounds of different nature. For example, we have flavoprotein. Flavoprotein is derived from vitamin riboflavin. So flavoprotein is one among the molecules present in the electron transport chain, as I showed you in the first diagram. So we have a flavoprotein mononucleotide, one among the examples of flavoprotein present in the electron transport chain. Then after flavoprotein, which can be abbreviated as FMN, you have coenzyme Q derived from vitamin E, and then you have group of iron containing pigment called cytochromes. Kwa hiyo electron transport chain imekuwa made up na nani? Flavoprotein, and then tuna nani? Tukitoka kwenye flavoprotein tuna nani? Tuna coenzyme Q. Toka hapo tuna cytochromes. Kwa cytochrome kama tulivyoona pale mwanzoni, kuna cytochrome B, cytochrome C, na tutaenda kuona nyingine huko mbele. So these molecules of electron transport chain are fixed in position within the inner mitochondrial membrane in such a way that they can pick up electrons from NAD and FAD-H2, that is reduce the NAD and reduce the FAD, and transport them in a definite sequence and a direction. So these molecules of electron transport chain zenyewe ziko kwenye inner mitochondrial membrane, na ndio maana tulikwambia kwenye adaptation kwamba the inner mitochondrial membrane inakuwa ina crystal, na zile crystal zinaongeza surface area for electron transport chain. That means kwenye zile crystal kuna hizi molecules za electron transport chain zimejifix mle. Zinavyojifix zinafanya transport of electrons and the protons, and at the end of the day tunapata energy in form of ATP kwa kule kutransfer hydrogen pamoja na electron. So in aerobic respiration, reduce the NAD and reduce the FAD become oxidized by transferring the pair of hydrogen atoms to the electron transport chain. Baadhi ya document tunasema kwamba hapo umeambiwa kwamba by transferring the pairs of hydrogen atom to the electron transport chain, lakini baadhi ya vitabu unaweza kukuta wamekuandikia the pair of electrons, sio pair of hydrogen atoms. Kimsingi ni kwamba FAD na NAD wana transfer pair of hydrogen atoms, lakini ile hydrogen atoms kule mbele inaenda kudissociate, so they dissociate later to form the hydrogen ions which are protons and the electrons. So the protons are not transported together with the electrons kwenye electron transport chain. Kwa hiyo tunaposema pair of hydrogen inakuwa carried by reduced NAD pamoja na reduced FAD, lakini at the end of the reaction, at the end of the day ni kwamba hiyo reduced NAD pamoja na reduced FAD zikifika kwenye electron transport chain, ile hydrogen inaweza kudissociate, then proton high transfer pamoja na electrons. So normally it is called as electron transport chain because nimekuonyesha pale mwishoni kwamba cytochrome B, cytochrome C, they transfer electrons only, not with the protons. So the hydrogen atoms attached to the hydrogen carriers, which are NAD and FAD, are transferred to a chain of other carriers at a progressively lower energy level. So what is the unique feature of electron transport chain is that the chains, they are arranged at a progressively lower level. So when hydrogen tend to transfer from one level to another, when we are saying about the progressively lower level, we are meaning that let's say here we have the series of hydrogen carriers and we are drawing them as if they are in this direction. That means if here we have NAD, let's say we have our NAD here, and when this NAD tend to move down the electron transport chain, it tend to release out ATP. It tend to release out ATP, and for the case of NAD, it releases 3 ATPs. While FAD, because FAD enters somewhere in the electron transport chain, let's say here, when FAD enters here, it releases only 2 ATP instead of 3 ATP is how NAD does. So what we are saying ni kwamba the hydrogen atoms attached to the hydrogen carriers, which are NAD and FAD, are transferred to a chain of other carriers at progressively lower energy levels. If the hydrogen pass from one carrier to the next, the energy released is coupled to the formation of ATP. So energy ambayo inakuwa released hapo hydrogen zinapopita, tunasema inakuwa coupled. Coupled maana yake ni kama inakusanywa. So the energy is accumulated to the formation of ATP. That means if the energy is not sufficient to form ATP, will be lost as heat. Kama ambavyo tumediscuss kule mwanzoni kwamba formation of ATP requires 30.6 kilojoule per mole. If we want to form 1 mole of ATP, it requires 30.6 kilojoule per mole. So combination of ADP with the inorganic phosphate to form ATP requires to form 1 mole of ATP requires 30.6 kilojoule of energy. Kwa maana yake hiyo energy ndio inakuwa coupled. Coupled ni kama unakusanya kitu afu unakifanyia kitu kimoja. Kama ukikusanya labda hela unazifanyia kitu kimoja baadaye ni kama tunasema zile hela umezikusanya. So the series of carriers is termed the respiratory chain. So that's why sometimes baada ya kuita electron transport chain tunaita respiratory chain. The carrier in the chain is as mentioned above. Kwa carrier kwenye chain tume-mention hapo juu kwamba inaanza flavoprotein, inafuata coenzyme Q, ikitoka coenzyme Q inafuata cytochromes, cytochrome B, cytochrome C. Kwa hiyo hiyo ndivyo series ambavyo ipo. So initial hydrogen atoms are passed along the chain, but these later split into the protons and electrons, and only the electrons pass from carrier to carrier. Initially tumesema nini? Initial hydrogen atoms are passed along the chain initially, but these later split into the protons and what? Electrons, and only electrons pass from carrier to carrier. For this reason, the pathway can be called the electron or hydrogen transport chain. So sometimes, some people they can call it the electron transport chain, while the others can call it the hydrogen transport chain, because kuna kipande ambacho ina transfer hydrogen na kuna kipande ambacho ina transfer electrons. Kwa hiyo majina yote mawili ni correct. However, the electron transport chain ndilo ambalo linatumika sana. Jina la electron transport chain ndilo ambalo linatumika sana kuliko jina la hydrogen or proton transport chain. Now, here we have a detailed mechanism, or we have a detailed step-by-step description of how electron transport chain tend to take place. However, my diagram is somehow large, so you will just tolerate. I will explain it very slow, and I'm sure you will understand what I mean and what we wanted to discuss in this part. Now we are saying that at the end of the chain, the proton and the electron combine, and the hydrogen atoms created link with oxygen to form water. Kwa hiyo kule mwishoni chain ikishaisha, pale tuliseparate proton na electron, kule mwishoni chain ikisha, tuna combine tena proton na electron, tunaenda kupata nani? Hydrogen atoms. Unaona? Very simplified mechanism of electron transport chain. Tukishapata hydrogen atoms combine with oxygen to form water. So oxygen ina part kubwa kama 2 au 3 kwenye respiration. Kwanza inatumika kwenye link reaction, kuweza kuondoa ile carbon dioxide kwenye decarboxylation reaction. Lakini pili, oxygen kwenye Krebs cycle, ikiwa kwenye form ya water ndio inayo supply oxygen kwenye Krebs cycle. Oxygen pia inafanya decarboxylation reaction pia kwenye Krebs cycle. Lakini jambo la tatu ambalo oxygen inafanya, ina act as the final hydrogen acceptor in the electron transport chain. So tunachokisema ni kwamba this formation of ATP through oxidation of hydrogen atoms is called oxidative phosphorylation. Is formation of ATP through oxidation of hydrogen atoms. Kwa nini tunasema oxidation of hydrogen atoms? Kwa sababu tunachukua hydrogen atom, tuna remove nini? Electron. Tuna remove electron. So removal of electron is called oxidation, while addition of electron is called reduction. So kuna diagram hii hapa ya electron transport chain, how it happens in detail, and this can be referred to human physiology by Stewart, Ira and Fox, page 108, figure 5.9. Now I don't have this book in my computer. If I could have it, I could show you, but I will find other books which I can show you the details of the electron transport chain. No problem. I have a lot of physiology books in my computer. Now what I want to explain to you is the detailed mechanism of how the process happens. I explained it in introduction, but I hope you didn't understood everything. Now tunavyokuwa tuna substrate, this shows what happens in Krebs cycle and glycolysis. So tunavyokuwa tuna substrate, let's say we have our substrate. Let us choose a color which can be seen well here. Let's say let it take red. Let it take red. Now let's continue. Tunapokuwa tuna substrate hapa anapokuwa na hydrogen, manake hii kwenye Krebs cycle au kwenye glycolysis, what happens ni kwamba huyu substrate anapokuwa anapita hapa anakutana nani na hydrogen carrier ambaye ni nadi. So hapa kinachofanyika ni kwamba hydrogen carrier anachukua hydrogen anaenda kutengeneza hii hapa ambayo ni nani, ni reduced nadi. Kwa kinachofanyika hapa, substrate hapa anakuwa oxidized by removal of hydrogen. From there hapa kwanza hii reaction ya kutoka hapa kuja hapa kumtengeneza huyu reduced nadi, hapa tunaweza kuseparate tukapata 2 ATP. Hizi ATP tukazidirect ziende hivi chini, lakini during this reaction it is coupled, energy produced here is coupled to formation of ATP. Kama unavyojua kwamba formation of ATP requires energy, kwa maana yake hapo kuna energy inapatikana ndio inatengeneza nini, ATP ya kwanza hapo. Kwa hii ndio first ATP inayokuwa formed, kwenye electron transport chain inakuwa inapatikana kama hapo. Lakini kutoka hapo akiwa fadi haingii kwenye this level, fadi does not enter to this level. Kama electrons zimekuwa carried by fadi haziingii hapa, zinaingia kwenye level ya chini. Maanake fadi H2 ni huyo hapa, si unamuona huyu fadi H2 ndo anaingilia hapa fadi. Kwa fadi anakosa hii ATP moja. So what will happen to fadi huku chini? Maanake tutaenda kuona hizi process nyingine zote zinazoendelea huku chini kwa fadi na nadi zinafanana, except hapo juu tu. Hapo juu fadi na nadi ndio wanatofautiana. Now hapo tumesha form 1 ATP kwa kutumia nadi, tumeform 1 ATP. Kwa from there sasa tukija hapa, kama fadi akiingia hapa au kama si fadi, basi electrons zikitoka kwenye nadi zinapitia hapo kwenye nini, kwenye flavoprotein. Zikishapita kwenye flavoprotein zinaenda wapi? Kwa enzyme Q. Tulisema kwamba kishatoka kwenye flavin mononucleotide flavin mononucleotide unaenda kwenye nani, kwenye enzyme Q. Huyu ni wa pili hiyo electron carrier. Kwa enzyme Q akigain electron anakuwa oxidized, afu akipoteza electron anakuwa reduced kwa enzyme Q. Tukitoka kwa enzyme Q tunaenda nani, cytochrome B. Lakini unaweza kaona, you can realize kitu kimoja kwamba hizi za juu hizi kuna nini tumezinania hapo juu, hata kama uzioni kwa sasa, lakini zilikuwa kwenye level moja. Tukaseparate zile tu proton zikapita kwa nje mpaka down, zilizofika huku chini ndio zikaungana nani na electron, tukatengeneza hydrogen and this hydrogen ikaenda kucombine with oxygen ikaform water. Sasa ninachokisema ni kwamba unaziona hizi hapa, unaweza ukanote kitu hapa. Lazima uwe ni mtu wa kunoti, yani tunafanya vitu sio tu unapita bila kuona chochote. Hapa unaona hizi zote hizi ziko kwenye one energy level. Kwa electron anapokuwa anapita hapa, kwa sababu ziko kwenye one energy level, maana yake hapa kwenda hivi juu ni increase in energy. Kwa electron anapokuwa anashuka this direction anakuwa anafanya decrease in energy. Kwa kuna some of the energy is lost. Kama unakumbuka general chemistry ni nani? Ni Bohr atomic theory. Kwamba electron anapokuwa na jump from one energy level to another, lazima kuwe kuna association of gaining of energy. Kwa ile electron anapokuwa na fall kutoka kwenye energy level moja kuja kwenye energy level nyingine, lazima kuwe kuna loss of energy. So that means electron anapotoka kwa mfano level ile pale, hii ni level moja, electron anapotoka pale kushuka hapa kuna loss of energy. That's why there is production of ATP. Hapa maana kuna loss of energy kwa sababu kuna fall ya energy level. Lakini kutoka hapa hadi hapa, this is the same energy level, so there is no loss of energy. Lakini unaweza kuona tukishafika kwenye cytochrome B kuna nini? Kuna mteremko tena pale, electrons zinashuka. Zikishuka maana there will be loss of energy. This will be coupled to ATP, will be coupled to ATP. So this lost energy hapa itakuwa coupled kwenda kwenye ATP. Toka hapo unaenda kwenye nini? Kwenye cytochrome C. Kwa kwenye cytochrome C tena this is the ion containing cytochrome pia. Ukifika kwenye cytochrome C kinatokea nini tena? Hapa electrons zinakuwa transferred kwenda kwenye cytochrome A. Lakini unaona tena cytochrome C and cytochrome A, they are at the same energy level. So hakuna chochote ambacho kitatokea hapo kwenye cytochrome C na cytochrome A. Electron atapita. Lakini anapotoka cytochrome A kwenda kwenye cytochrome huyu hapa A3, anapotoka hapo kwenda kwenye cytochrome A3 kuna kushuka hapa. Kwa kushuka huko hapa kunatengeneza nini? ATP. There will be loss of energy. This energy which is lost here is coupled to the formation of ATP. Manake nakusanya hapa inatengeneza ATP. And then from there the electron is transferred to hydrogen. Then combination of a electron and a proton form the hydrogen, and then hydrogen later combine with oxygen to form water. So oxygen acts as the final final hydrogen acceptor in the electron transport chain. So this is the detailed mechanism, and as I told you, it is from the book of human physiology. We have a summarized mechanism of electron transport chain. Is how I explained to you before about this diagram. So I hope you still remember with this diagram, and I will not lose a lot of minutes here. Now in this diagram it is simplified, and it is taken from Understanding Biology book. So it is simplified. Unaweza kuona tunatoka kwenye NAD H2 tunapata nini? Flavoprotein kama kuna fad anaingiza pale, then but this cannot be learnt well. Kwa sababu gani? Huyu na huyu hawamuonyeshi kwa yuko kwenye level moja. So that means kama hapa kweli anashuka, angekuwa lazimika kupoteza energy. Hii haina description nzuri kuliko ya mwanzo ambayo imeeleza vizuri zaidi. Unaweza kuona hizo cytochrome B, cytochrome C, cytochrome A, na hii sio detailed sana kama ile ya mwanzo. Kwa hizo ni diagrams tu of electron transport chain. Ni diagrams ambazo zina describe the mechanism of electron transport chain. So yeah, I think you have already understood about the basic concept of electron transport chain. Now we can go to discuss about the concept of oxidative phosphorylation, how ATP is produced from oxidative phosphorylation, and from there we'll move to relationship between electron transport chain and Krebs cycle. Also we'll move to the details of the electron transport chain. Now let's discuss about the oxidative phosphorylation. We discussed in nutrition in plant about the concept of photosynthetic phosphorylation, and in photosynthetic phosphorylation or photophosphorylation what we discussed is that light energy is used in production of ATP. Light energy is used in production of ATP, but here it is the energy from oxidation which is used in the production of ATP. So this is the process of forming ATP from ADP and inorganic phosphate using energy from oxidation of reduced hydrogen carriers. So we have reduced hydrogen carriers which are reduced in NAD and reduced FAD. We are oxidizing them. So we are carrying that hydrogen, we are oxidizing them, because the removal of hydrogen is oxidation. So by removing hydrogen from them, it is oxidation. So by oxidizing them, that hydrogen is transferred in the series of different carriers, and by transferring that hydrogen, we are getting energy. So we are saying that for each reduced NAD, that interrespiratory chain, 3 ATP can be made as hydrogen or electron flow to oxygen. So when a NAD enters the electron transport chain, each NAD produces 3 ATP. However, for each reduced FAD, only 2 ATP are made because reduced FAD enters the chain at lower energy level. Nime-explain hichi kitu kwenye diagram ambayo ilikuwa hapa kwamba FAD inaingia at lower energy level. Unaweza kuona hapa jinsi ambavyo inaingia, lakini ukiona hiyo uielewe, basi ile diagram ya mwanzo ndio ilikuwa inaeleza vizuri zaidi. Unaweza kuona hapo nini ambacho kinakuwa kinaonekana. NAD anaingia kwenye level hii hapa. Huyu ndio NAD. Lakini FAD anaingia kwenye level hii hapa. So this is the lower energy level ukilinganisha na hii, kwa sababu nimekuambia huku tunaenda wapi? Increase in energy level. Tunapanda juu, increase in energy level. So FAD anaingia kwenye lower energy levels compared to NAD. So hiyo ndo mechanism ambayo inakuja kusema kwenye notes kwamba it is because of the difference in the energy level ambazo hizi molecules zinaingia. Kwa tunachokisema ni kwamba the overall equation for respiratory chain tunaandika hivi: 12 hydrogen 2 plus 6 oxygen, tunapata 12 water plus 34 ATP. So unaweza kuona electron transport chain ndio inayotengeneza ATP nyingi zaidi. Manake 1 molecule of glucose metabolism, complete metabolism of 1 molecule of glucose produces 38 ATPs. Lakini 34 zote zinatoka wapi? Electron transport chain. Na ndio maana tutaenda kusoma huko mbele kuhusu cyanide. How cyanide inaweza kupelekea cell death. Kwa sababu cyanide inaenda ku-inhibit the components of electron transport chain. Na ukumbuke hapa electron transport chain inatengeneza ATP ngapi? 34. Kwa kama inatengeneza ATP 34, that means ATP nyingi zinatoka kwenye electron transport chain. Akija cyanide akabreak the formation of this ATP, manake only 4 ATP will be produced, 2 from glycolysis and 2 from Krebs cycle. So at the end of the day utajikuta una few ATP and then cell inakufa because of the deficiency of energy. Manake energy does not meet the requirements. Kwa hiyo kwenye glycolysis and Krebs cycle inakuwa glucose plus 6 water. Glycolysis and Krebs cycle unapata 6 carbon dioxide plus 12 hydrogen plus 4 ATP. Lakini kwenye electron transport chain 12 hydrogen hizi hapa zinaenda wapi? Zinaenda huku. Zinakuwa carried by NAD and FAD. Hazikifika huku NAD and FAD zinaenda oxidation. So 12 hydrogen plus 6 water. In the respiratory chain we are getting 12 hydrogen and oxygen plus 34 ATP. So tukiadd equation 1 and equation 2, manake glucose, water, carbon dioxide, 12 hydrogen, 4 ATP. Tukiadd kutapata, manake unaona kuna 6 water, kuna 12 water. So 6 water itaisha. Hapa tutabakia na ngapi? Tuna 6 water. Lakini pia 6 oxygen hapa itabakia. 6 oxygen itabakia. 12 hydrogen na 12 hydrogen zitaenda na maji, So tutabakia tuna glucose plus 6 oxygen, tutapata 6 carbon dioxide plus 6 water. 6 carbon dioxide plus 6 water plus 38 ATP. Thus 38 molecules of ATP are produced for every glucose molecule oxidized in aerobic respiration. Kwa hiyo molecule 38 za ATP zinatengenezwa kwa kila glucose. Glycolysis ni 2, Krebs cycle ni 2, electron transport chain ni 34. Zinakuaje 34? Tutaenda kuona calculation huko mbele, lakini calculation yake ni very simple kupata hiyo 34 ATP. So the rule of oxygen is to act as the final hydrogen atom acceptor. While in many stages in respiration, oxygen is nevertheless vital as it drives the whole process. Kwa hiyo oxygen tunavyosema ina act as the final hydrogen acceptor, hatuna maana kwamba haitaki kwenye steps mbalimbali. Nimekuambia kwenye link reaction oxygen inatumika kwa ajili ya decarboxylation. Kwenye Krebs cycle oxygen from water inatumika kama ambavyo tu discuss kwenye previous session. Kwa hiyo oxygen sio kwamba haina umuhimu. Oxygen ina umuhimu mkubwa tu, ina umuhimu mkubwa, lakini ukiulizwa what is the role of oxygen in respiration, unasema kwamba ina act as the final hydrogen atom acceptor. So in its absence only anaerobic glycolysis can continue. In its absence only anaerobic glycolysis can continue. So in the absence of oxygen you can't do Krebs cycle and you can't do electron transport chain. So oxygen is very important in the whole process of respiration because of how it tend to allow different reaction of respiration to take place. Now let's continue with our discussion about the oxidative phosphorylation. So the transfer of hydrogen atoms, the transfer of hydrogen atoms to oxygen is catalyzed by the enzyme cytochrome oxidase. So hiyo cytochrome oxidase iko kule mwishoni, and this enzyme is inhibited by cyanide. Very important point. Yani ukikumbuka usisahau hichi kitu. Cytochrome oxidase is inhibited by nini? By cyanide. Cyanide inaandikwa kama CN. Huyu mdudu sio mzuri. Kama umeshaisikia Wachaga hawalagi mihogo ni kwa sababu ya cyanide. Mihogo mingine huwa inakuwa na cyanide. Kwa hiyo ikiwa na cyanide unakufa ukila. Sio tu Wachaga yeyote, lakini wao ilikuwa ni mila kwao kwamba sisi hatuli mihogo. Kwa sababu gani? Kwa sababu ya cyanide. Cyanide sio mzuri huyu. Kwa cyanide yeye akienda kuinhibit huyu hapa. Very enjoyable concept. Yani cyanide akienda kuinhibit cytochrome oxidase, maana yake zina accumulate electrons and protons they accumulate. So the accumulations of the protons and electrons zinasababisha hata stages nyingine the electron transport chain zishindwe kuproceed. So that accumulation of the protons and electron will lead to the stop of the process of the electron transport chain. Kwa process nzima electron transport chain itafanyaje? Ita stop. Ikisha stop, hence cell itafikia wakati ambao itakosa energy, itadai. Kwa hiyo ndo sababu Wachaga hawali mihogo. Kwa sio kasema ni uchawi. Sio uchawi, it is because of the inhibition of cytochrome oxidase by cyanide. So preventing the removal of hydrogen atoms at the end of respiration chain. Kwa kama ikiwa inhibited by cyanide, ina prevent removal of hydrogen atoms at the end of respiratory chain. In these circumstances, the hydrogen atoms accumulate and the aerobic respiration cases, making cyanide a most effective respiratory inhibitor, na anapelekea kufa kwa seli, hence kufa kwa organism mzima. Very enjoyable concept for you to know it. Very enjoyable concept for you to know about cyanide poisoning. Kwa unavyoenda maabara ukisikia sauti yote na cyanide, iogope sana. Hiyo ukilamba au kwa bahati mbaya ukimeza sauti yote yenye cyanide, hiyo cyanide ikifika kwenye seli, kwenye electron transport chain, itakachofanya hautaamini. Tunaweza tukakuzika siku ya pili yake tunaweza kukuzika. Hayo ndio madhara ya cyanide. So tuangalie kidogo kuhusu relationship kati ya electron transport chain na tricarboxylic acid cycle, yani Krebs cycle. Kwanza Krebs cycle au TCA produces reduced hydrogen carrier such as FAD and reduced NAD. These carry hydrogen atoms which are changed to metabolic energy in electron transport chain. Kwa tunaposema relation kati hizo mbili, hapo usicomplicate vitu, ni very easy. Usicomplicate, don't complicate anything. You know, if you complicate, at the end of the day utaishia kufeli hata mitihani. Unaangalia tu kwamba je, Krebs cycle na electron transport zinahusianeje? Kwamba Krebs cycle inatengeneza reduced NAD na FAD zinazoenda kutumika kwenye electron transport chain. Lakini jambo la pili, both Krebs cycle and electron transport chain release ATP as the end product. So unaona vitu ni very easy, yani havihitaji hata complicate, Kwa hiyo sasa baada hapo tunaenda kwenye nini? Kwenye electron transport chain in detail. Tunaenda kuchimba ndani. Hapa tumegusa juu. Kwa wale wenzangu na mimi ambao ni wavivu, ishieni hapo. Ila kwa wale ambao wanataka kuwa deep, unajua nikwambie kitu kimoja ambacho ni very enjoyable, mimi nikitumia sana kwenye kusoma. Nilipokuwa nasoma hivi vitu afu nakuwa deep, nilikuwa siangai kwenye mtihani yani. Very easy. Nakuwa na concept mengi, nikishakuwa na concept mengi, at the end of the day najikuta tu ni mtu ambaye tayari yani vitu vingi nashuka tu yani kirahisi sana. Kwa hiyo kwa wale ambao mnataka kuwa deep na muweze kushuka vitu kirahisi, wewe niwape tu break kwanza ya dakika chache, alafu nitakurudia tuendelee na hiyo electron transport chain. Hiyo ni reference kutoka The Cell a Molecular Approach, page namba 70. Hicho kitabu nyinyi mkisome, na ndio maana nimeambia hapa tunachimba. Wenzangu na mimi wale wa kuishia BS in understanding, kipindi kimeishia hapo, lakini wale ambao mnataka kuendelea, kipindi bado kinaweza kuchukua hata masaa mawili. Kwa hiyo break kidogo, kunywa maji, pumua, stop video afu tutaendelea. Now let's continue with our session. I have this book in my computer, so it is in the page 90, not 70 as how it is documented here. Now we are saying that the coupling of electron transport chain to production of ATP, coupling of electron transport chain to production for ATP. However, what is explained in the book, it is somehow different from what is present here, but you shall see the detailed mechanism of how ATP is produced. So we are saying that the lollipop-like structure can be seen protruding from the inner mitochondrial membrane into the matrix when viewed with electron microscope. We have the lollipop-like structure. The cristae are also covered with this structure. The lollipop-like structures are called respiratory assemblies and are critically important in the generation of ATP. Sometimes instead of being called as respiratory assemblies, they are called as the elementary particles. So the lollipop structures, they are important in the coupling of energy in the formation of ATP. So we are saying that the Chemiosmotic Theory is the most widely accepted explanation of how the electron transport chain produces energy. Chemiosmotic Theory. The Chemiosmotic Theory. So what it explains, we shall see. However, it is just in a few details, because if we give you many details, we'll end up at confusing you. So we are saying that the Chemiosmotic Theory, the most widely accepted explanation of how the electron transport system produces ATP. How electron transport system produces ATP. According to the Chemiosmotic Theory, 3 members of electron transport system become active proton pump when they accept a pair of electrons. 3 members of electron transport chain become active proton pump. So zina pump proton kwenda wapi? When these members become reduced, they move a pair of proton from the matrix to the space between the inner and the outer mitochondria membrane, ambayo tuliita ni intermembrane space. Note that for every transported electrons that originate from NAD, 6 protons will be pumped. Since FAD donate electrons further down the line in the electron transport system, electrons from FAD activates only 2 of 3 protons pump. We shall see from the book which are the 2 protons pump activated by FAD. Kwa hiyo As the result of electron transport and activation of protons pump, the hydrogen ions concentration in the space between the inner and outer membrane is much higher than in the matrix. Kwa tunatengeneza nini? Tunatengeneza a steep electrochemical gradient is thus established across the inner mitochondria membrane. So the lollipop-like respiratory assemblies consist of a group of proteins that form a stem and a globular subunit. So the stem contains a channel through the inner mitochondrial membrane that permits the passage of protons and hydrogen. The globular subunit, which protrudes into the matrix, contains an empty synthase, an ATP synthase enzyme that is capable of catalyzing the reaction ADP plus phosphate to form ATP. So this is the explanation of the detail about the electron transport chain and the Chemiosmotic Theory, which suggests how ATP is produced during electron transport chain. Kwa hiyo tunakuwa tuna protein tatu ambazo zina act kama proton pump, zikipump proton. Hizo protein zikipump proton zinaongezeka kwenye intermembrane space. Then after there tunakuwa tuna nini? Tunakuwa tuna a lollipop-like structure ambayo inakuwa ina hole pale katikati kwenye stem, lakini kule mwishoni ina nini? Ina 8 phase ambayo ina catalyze formation of ATP. So ninachokisema ni kwamba the inner mitochondrial membrane is permeable to hydrogen. Is not. So the inner mitochondrial membrane is not permeable to hydrogen. However, the hydrogen ions can diffuse through the channels provided by the stems of the respiratory assemblies. So kwa sababu inner mitochondrial membrane ni impermeable, manake kule kwenye channel peke yake ndo ambao kutapitisha hydrogen. As each pair of hydrogen move into and through the bulbous portion of respiratory assemblies, the ATP synthase enzyme in this portion is activated and a molecule of ATP is produced as each pair of hydrogen ions. Kwa hiyo kwenye nadi zinakuwa pumped how many? Zinakuwa pumped 6. Kama 6 maana kita produce how many? 3 ATP. NB, the energy provided in the formation of ATP is derived from the transition of electrons, that is oxidative phosphorylation. Kwa hiyo even ndivyo ambavyo Chemiosmotic Theory inakuwa ina suggest, since each node result in the pumping of 3 pairs of protons by electron transport system or chain, 3 ATP are produced. Since FAD result in the pumping of 2 pair of proton, 2 ATP are produced as the protons diffuse back into the matrix through the respiratory assemblies. Kwa hichi ndicho ambacho nilikuwa nakielezea hapa. Unaweza kuona hapa kuna matrix ina low concentration of hydrogen, lakini akija huyu nadi akija kwa mfano huyu nadi hapa. Kwa hiyo unaweza kuona hapa na tunaanzia hapa. Explanation yetu tunaanzia hapa. Kwa tukianzia hapa huyu nadi, huyu maana yake ndio nadi hapa. Anampa huyu hapa electron. So maana yake from here hii electron inakuwa inapita huko ndani kwenye series of nini, electron carrier. Lakini wakati electron inapita ndani kwenye series of electron carrier, huko ndani we have proteins which pump hydrogen kuja huko. So proteins ina pump nini, hydrogen kuja huko. Tuna protein nyingine tena pale na pump hydrogen kuja. So we'll have how many? We'll have 1 hydrogen, I mean 1 pair, second pair, third pair, first pair, second pair, third pair of hydrogen. This is the space between membrane, because this is the inner membrane, this is the outer mitochondrial membrane. Membrane kama unavyojua ni phospholipid bilayer. Phospholipid bilayer maana yake hapa tuna phospholipid na huku pia tuna phospholipid. So hii hapa ni inner, hii ni intermembrane space. From there maana tunakuwa tumegenerate nini? Very important for you to remember, tumegenerate hydrogen ions gradient. Tumegenerate hydrogen ions gradient. So tuna many hydrogen ions this side as compared to this side. So hii na mitochondrial membrane ni impermeable to hydrogen. So maana hydrogen zina njia moja tu ya kupita kurudi kule, which is this one. So hydrogen ni pass, lakini zinapopita pale hapa kuna enzyme 8 phase. Kwenye hii kuna 8 phase ambayo inakataza formation of ATP. So that's why at the end of the day tunafika kule mwishoni ambako tunakuwa tunatengeneza ATP, tunakuwa tunatengeneza ATP. Lakini I want to show you the book and I hope utaelewa zaidi hicho ambacho nakielezea. So this is our book. Na na hivi ndivyo ambavyo unaweza ukaona. Sasa nataka nielezee kidogo kuhusu kile ambacho kilikuwa kinaelezwa kule. Kwa huku kwenye kitabu wameeleza maelezo mengi kidogo as compared na kule kwenye notes, lakini na kushauri tu maelezo kwenye notes yale yanakutosha for your level. So I will explain some of the few details ili uweze kunielewa, lakini that is how electron transport chain inatokea kwenye detail. So tunavyosema ni kwamba most of the usable energy kutoka kwenye Krebs cycle na glycolysis inakuwa bado iko kwenye nadi. So most of the usable energy obtained from breakdown of carbohydrates or fat is derived by electron transport and oxidative phosphorylation. For example, the breakdown of glucose by glycolysis and TCA yield a total of 4 molecules of ATP, 10 molecules of NAD and 2 molecules of FAD. That means tunatengeneza 10 molecules of FADH na 10 molecules of NADH and 2 molecules of FADH. So hizo 10 molecules of FADH and 2 molecules of FADH zinaenda kuuliza how many? 34 ATP kama tulivyosema kule mwanzoni. 34 ATP in the electron transport chain, 34 ATP. So adding 34 plus 4 ATP from Krebs cycle and glycolysis directly, we are getting 38 ATP. Lakini that's not the important thing about tutaka tueleze. So the electrons from NAD and FAD are then transferred to the molecular oxygen, which is coupled to the formation of additional 34 ATP molecules by oxidative phosphorylation. The transfer of electrons from NAD to oxygen releases a large amount of free energy, about negative 52.5 kilocalories per mole for each pair of electron transferred, so that this energy can be harvested in usable form. The process takes place gradually. And we can move to our next page. By the passage of electrons. So the process takes place gradually by the passage of electrons through a series of carriers which constitute the electron transport chain. The components of electron transport chain are organized into 4 complexes. Now this is what I wanted you to know. Kutoka kwenye notes kule yameandikwa maelezo, lakini nataka nikuonyeshe na diagram ili uweze kujua kile ambacho tuko naeleza nini. Kwa components za electron transport chain are organized into 4 complexes in the plasma membrane of aerobic bacteria and in the inner mitochondrial membrane of eukaryotic cell. Kwa kama kwenye mitochondria, kwenye inner mitochondrial membrane ndio tunakuwa na components za electron transport chain. Kwa hiyo NAD is produced from TCA or citric acid cycle in mitochondrial matrix. Electrons from NAD then enter the electron transport chain in complex 1. Complex 1 which constitute 44 polypeptide chain in eukaryotic cell. Electron from NAD they enter at complex 1 which constitute of 44 polypeptide in eukaryotic cell. So from there nataka tu tuweze kuelewana kirahisi kuhusu hichi ambacho nakuwa nakielezea. Tuliona kirahisi kuhusu ambacho nakielezea. Kwa complex 1 ni polypeptide. Unaona? Hizo ni electron from NAD. These electrons are initially transferred from NAD to flavin mononucleotide. They are transferred from NAD to flavin mononucleotide FMN, ambayo tu discuss kule nyuma, and then through a series of iron sulfur carrier to coenzyme Q. Unaona, tukitoka kwenye flavin mononucleotide tunafuata nani? Coenzyme Q. Kwa hapo katikati kuna vitu vingi, lakini inayofuata nani? Coenzyme Q. An energy yielding process with change in energy of negative 16.6 kilocalories per mole. Kwa hizo electrons zinapokuwa transferred from flavin mononucleotide kwenda kwenye coenzyme Q, tuna release energy of 16.6 kilocalories per mole. From there tunachokisema ni kwamba coenzyme Q is sometimes known as ubiquinone. So from there, ubiquinone is the small lipid soluble molecule that carries electrons from complex 1 through membrane to complex 2. You see, tunatoka complex 1, coenzyme Q anapeleka wapi? Complex 2, which constitute of about 10 polypeptide. Complex 1 ilikuwa na 44, complex 2 ana about 10. In complex, no, it is not from to complex 2, it is to complex 3. Hapa kwenye NAD akitoka kwenye ubiquinone, that is coenzyme Q, anaenda kwenye complex 3 moja kwa moja. Tutaenda kuona complex 2 anafanya kazi gani. Kwa hiyo tukisema which constitute of about 10 polypeptide. In complex 3, electrons are transferred from cytochrome B to cytochrome C. Complex 3, electrons are transferred from cytochrome B to cytochrome C. An energy yielding reaction with a change in G of about negative 10.1 kilocalories per mole. You see, energy yielding reaction ya kwanza ni hapa, electron transfer kutoka kwenye flavin mononucleotide kupita kwenye series ya iron sulfur carrier to coenzyme Q, negative. Lakini energy yielding reaction pia nyingine ni hapa. Tunaweka negative kwa sababu gani? Ni exothermic, that means ina release energy. Kwa reaction ambazo zina release energy kuliko zile ambazo zinatumia, hizi zina release. From there we have cytochrome C, then carries electron to complex 4. Unaona? So cytochrome coenzyme Q anampa complex 1. Kutoka complex 1 napita through membrane, coenzyme Q anampa complex 1. Complex 1 through membrane anachukuliwa na huyu coenzyme Q kwenda kwenye complex 3. Ukitoka hapo unaenda kwenye complex 4. So cytochrome C then carries electron to complex 4, which is cytochrome oxidase. So complex 4 ndio ile enzyme cytochrome oxidase, ambayo if you remember tulisema anakuwa affected na nani? Na cyanide. Cyanide anaenda kuvunja complex 4, anaenda kuinhibit. Akinhibit, that means inhibit the process of respiration, where they are finally transferred to oxygen. So they are finally transferred to oxygen, na hiyo transfer change in energy negative 21.8 kilocalories per mole. The energy yielding electron transfers at each of these complexes is coupled to the synthesis of ATP. Now, tunafahamu kwamba energy yield inakuwa coupled to the synthesis of ATP. But the question come, how synthesis of ATP occur? Ndiko ambako tunarudi kwenye theory yetu ambayo discuss, ambayo ina suggest the formation of ATP from Chemiosmotic Theory, ambayo suggest for the formation of ATP. Na tunachokisema ni kwamba kuna complex nyingine ambayo ni complex 2. Complex 2 yeye anachukua nani? Anachukua fadi. Complex 2 anachukua fadi. Akishamchukua fadi anamuingiza moja kwa moja mle ndani bila kupitia stage fulani. So a distinct protein complex 2 which consists of 4 polypeptide receives electron from citric acid cycle intermediate succinate. These electrons are transferred to FAD. Complex 2 anachukua electron toka kwa nani? Kwa succinate, anapeleka kwa FAD rather than NAD, and then coenzyme Q. Kwa electrons zinatoka kwenye complex 2 zinaenda kwenye FAD, zikitoka FAD zinaingia wapi? Zinaingia coenzyme Q moja kwa moja. Kwa hazipiti kwenye njia ya flavin mononucleotide, zinapita kwenye nini? Kwenye coenzyme Q moja kwa moja. From coenzyme Q, electrons are transferred to complex 3 and then to complex 4. Kwa unaona kutoka kwenye coenzyme Q anaenda complex 3 complex 4 is already described. Manake process different kutoka huku juu ambako kulikuwa kuna flavin mononucleotide is already described in contrast to the transfer of electron from NAD to coenzyme Q at complex 1. Kwa hiyo FAD haina kupitia kwenye complex 1. Kwa hiyo sasa nataka tuone kwanza nataka tuone tu vile ambavyo tulikuwa tunaeleza. Nataka tuvione hapa vile ambavyo tulikuwa tunaeleza kwenye haya macoenzyme na kwenye hizo complex. Nataka tuone kwenye diagram, in terms of diagram how it happens. Unaweza kuona hapo hii ni electron transfer kwa FAD, I mean kwa NAD. Sorry, this is not FAD, it is NAD. Kwa NAD unaona huyu ni complex 1, a transfer electron to complex 1. A transfer electron to complex 1 ana pump proton kuja kwenye intermembrane space. Huyu ni NAD, unaona NAD ana transfer electron, ana pump proton. Cytochrome Q huyo hapa. Cytochrome Q anapeleka wapi? Complex 3. Complex 3 4 hydrogen zinaenda kule. Cytochrome C anachukua hapo, anapeleka wapi? Anapeleka pale ambaye complex 4. Ambaye complex 4 tumesema ni cytochrome oxidase. Kwa cytochrome oxidase maana yake reaction hii inatokea inaenda kuform water. Hapa pia zinakuwa pumped proton. Hizi proton zote kuanzia hapa ukiangalia hii reaction maana yake hizi proton zote unaweza kuona tu okay. So hizi proton zote ukianzia hapa kuna proton hizi hapa. Hizi zote zinakusanywa zinaenda wapi? Pale. Zikifika pale zinapita kwa yule. Zikipita kwa yule kuna 8 pairs pale. Kwa zikipita kwa 8 pairs zinakuja kutoa nini? ATP. Zinakuja kutoa ATP. Kwa hiyo that's how things happen. Hakuna uchawi wote. Maana kuna kupump kwanza proton, process ya kwanza. Kuna pump proton kwenda kule. Kuna pump proton. Hiyo ni process ya kwanza ya kupump proton. We are first pumping the protons. Una pump na proton za hapa. Una pump na proton za hapa. Kwa hapa napo na pump ziende kule. Hapa napo na pump ziende kule, then at the end of the day hizo proton zote zinaenda kuingia kule. Kwa zikiingia kule ndo zinatengeneza ATP. So this is how kwenye NAD vitu vinakuwa. Hivyo ndivyo ambavyo kwenye NAD vitu ambavyo vinakuwa. Kwenye NAD vitu vinakuwa kwenye sura hiyo hapo. Kwa hiyo hii diagram labeling yake wanasema kwamba ni electron transport chain in mitochondria. This is the electron transport chain in mitochondria. So mitochondria are bounded by double membrane system. Bounded by double membrane system consisting of the inner and outer membranes. Fold of the inner membrane cristae extending to the matrix. NAD is produced from Krebs cycle in the matrix. Now electrons from NAD tunasema enter the electron transport chain at complex 1. Unaona zinaingia at complex 1. The electrons are then transferred to complex 3 by coenzyme Q. So coenzyme Q, then complex 3, coenzyme Q then coenzyme Q to enter complex 3, and then to complex 4 by cytochrome C. Cytochrome C, complex 4, where they are transferred to oxygen. So tunaenda kwenye oxygen kukupitia nani? Kukupitia complex 4 ambaye ni cytochrome oxidase. Cytochrome oxidase ambaye ni enzyme. So the electron transfer in complex 1, 2 and 4 are associated with a decrease in free energy. A decrease in free energy. Electron transport kwenye complex 1, 3 and 4 are associated with a decrease in free energy. So hiyo decrease ya free energy ndio ambayo inatumika kupampu proton sawa sawa na Chemiosmotic Theory. So which is used to pump protons from matrix to the intermembrane space. This establishes a proton gradient across the inner membrane. The energy stored in the proton gradient is then used to drive ATP synthesis as the proton flow back to the matrix through complex 5. So complex 5 ndio hii hapa ambayo protons zinavyokuwa zinatoka huko ndani zina flow kuja huku tena kwenye matrix kwa kupitia complex 5. So complex 5 maana yake ndio tuisema kuna stock na pia kuna kwa hii ime contain 8 pairs enzyme. Kuna stock na pia kuna hizo ndo tunaziita stocked particles. Lakini hiyo ni nadi. Now in fadi kama tulivyosema kwamba kuna complex 2. Complex 2 anachukua moja kwa moja anapeleka kwa nani? Anapeleka moja kwa moja kwenye cytochrome Q and hence complex 3. Kwa unaweza kuona hivi ndivyo ambavyo inatokea. Kwamba hapa tuna succinate. Tukiona succinate huyu hapa, nini kinatokea? Succinate anachukua electron kwa kupitia complex 2 zinaenda kwenye fadi. Fadi anampelekea nani? Cytochrome Q moja kwa moja. Complex 3 kwa anachukua hydrogen cytochrome complex 4 then complex 5. Kwa hiyo maana yake unaona hapa fadi atakuwa amepump proton chache ukikompare na nadi ambayo tuko tunamuona kwenye previous page. Kwa kama fadi amepump proton chache, unaona nadi ana pump proton hapa, ana pump hapa proton na hapa ana pump proton. Lakini fadi ana pump proton sehemu mbili tu. Kwa hiyo ndio maana fadi huwa anatengeneza ngapi? 2 ATP, not 3 ATP. So we are saying transport of electron from fadi, electron from succinate into the electron transport chain via fadi in complex 2. They are then transferred to coenzyme Q and carried to the rest of the electron transport chain as described in figure. So the transfer of electrons from fadi to coenzyme Q is not associated with the significant decrease in free energy. Transfer of electron from fadi to coenzyme Q is not associated with the significant decrease in free energy, and the free energy is produced only at complex 3 and 4 kwa sababu hiyo pumping of protons. Free energy is produced only at complex 3 and 4. Complex 3 and 4. So tunachokisema ni kwamba hayo ni maelezo ya nadi na fadi, lakini hatujaenda kuelezea the real mechanism how it happens. So the free energy derived from the passage of electron through complex 1, 3 and 4 drives the synthesis of ATP by oxidative phosphorylation. Free energy at complex 1, 3 and 4 drives synthesis of ATP. Kwa sababu yu fadi anahusisha complex 3 na 4 peke yake, atengeneza mbili wakati nadi atengeneza 3. Now importantly, the mechanism by which energy is derived from this electron transport reaction is coupled to ATP synthesis is fundamentally different from synthesis of ATP during glycolysis or citric acid cycle. Kwa hiyo lazima ujue hichi kitu kwamba tunavyotengeneza ATP kwenye electron transport chain ni tofauti na kutengeneza ATP kwenye glycolysis au kwenye Krebs cycle. Ni tofauti na kutengeneza ATP kwenye glycolysis au kwenye Krebs cycle. So kwenye glycolysis pamoja na Krebs cycle, ndio maana nasema in later cases ambazo ni glycolysis or Krebs cycle, a high energy phosphate is transferred directly to ADP from the other substrate of energy yielding reaction. Kwa mfano, kwenye final reaction of glycolysis, in the final reaction of glycolysis, the high energy phosphate of phosphoinopyruvate, is transferred to ADP yielding pyruvate plus ATP. Unaona? Hiyo ni kwa mfano phosphoinopyruvate. Lakini kama unavyojua kwamba we have two stages in glycolysis ambazo zinatengeneza ATP. Kwa phosphoinopyruvate ni mfano. Mfano mwingine ni 1.3 diphosphoglycerate. Kwa naye maana yake tutachukua ile phosphate ya kwenye kaboni ya kwanza, tutampa ADP, phosphate ya kwenye kaboni ya tatu tutamwacha katika kutengeneza ATP. Kwa in Krebs cycle na glycolysis tuna remove phosphate kutoka kwenye a high energy intermediate, tunampa ADP. Lakini kwenye electron transport chain ni tofauti. Such direct transfer of high energy phosphate group does not occur during oxidative phosphorylation. Instead, the energy derived from electron transport is coupled to the generation of a proton gradient across inner mitochondria membrane or bacterial plasma membrane. Kwa energy ambayo inapatikana pale, energy derived from electron transport, vile ulivyo transport electron unapata ile energy yenyewe inatumika tu kupump proton. So ile energy haigenerate ATP moja kwa moja, bali ina pump proton. After pumping the proton, then zile protons zatengeneza gradient. The potential energy stored in this gradient is then harvested by a fifth protein complex, that is protein complex 5, which couples the energetically favorable flow of proton back across the membrane to the synthesis of ATP. So the theory behind this is what we call as the Chemiosmotic Coupling or Chemiosmotic Theory ya kutengeneza ATP kwenye nini? Kwenye electron transport chain. So nachokisema ni kwamba the mechanism of coupling electron transport to ATP generation, that is Chemiosmotic Coupling, is a striking example of relationship between structure and function in cell biology. Structure and function in cell biology. So you have the stocked particles with the hollow structure, and because of the impermeability of the inner mitochondrial membrane to proton, that enable the proton to pass only enable the protons to pass only in the protons, they pass only at the stocked particles, and then at the end of the day we'll end up with the production of ATP. So the hypothesis of chemiosmotic coupling was first proposed in 1961 by Peter Mitchell, who supposed that ATP is generated by the use of energy stored in the form of proton gradient across biological membranes rather than by direct chemical transfer of high energy groups. And this is actually what happens, is how we explained kule kwenye notes. Kwa hiyo this is not a direct transfer of phosphate from high energy molecule, but it is by the coupling. It is by the production of the proton gradient ambayo itakuja kufanya coupling. Now, biochemists were initially highly skeptical of this proposal, and the chemiosmotic hypothesis took more than a decade to win general acceptance in the scientific community. Overwhelming evidence eventually accumulated in its favor. However, under the chemiosmotic coupling is now recognized as the general mechanism of ATP generation operating not only in mitochondria and bacteria but also in chloroplasts during photosynthesis. So unaweza kuona kwamba hiyo nini chemiosmotic coupling jinsi ambavyo inatokea. Kwa chemiosmotic coupling tumeelezea kule, tumeelezea unaweza kuona tu kwamba electron transport through complex 1, 3 and 4 is coupled to the transport of proton out of the interior of the mitochondria. Tumeshaeleza. Thus energy yielding reaction of the electron transport establishing a proton gradient across inner mitochondrial membrane. Complex 1 and 4 act as proton pumps that transfer protons across the membrane as a result of conformational changes induced by electron transport. In complex 3, protons are carried across membrane by coenzyme Q, which accepts protons from the interior of the mitochondria at the complex 1 or 2 and releases them on outside of the membrane at complex 2. Lakini zote muhimu cha muhimu wewe kujua ni energy inakuwa formed vipi. Complex 1 and 3 each transfer 4 protons across the membrane per pair of electrons. In complex 4, 2 protons per pair of electrons are pumped across the membrane and another 2 protons per pair of electrons are combined with oxygen to form hydrogen with mitochondria. Thus, the equivalent of 4 protons per pair of electron is transported out of mitochondria at each of these 3 complexes. This transfer of protons across mitochondrial membrane play the critical role of converting the energy derived from oxidation reduction reaction of electron transport to the potential energy stored in the proton gradient. Because protons are electrically charged particles, the potential energy stored in the proton's gradient is electric as well as chemical in nature. Now this is the key experiment of the Chemismatic Theory as done by the Peter Mitchell and other scientists which contributed, but I will not explain it. If you need this, you will just go to this book, A Cell Molecular Biology Approach. Then you can read this. You can read this from the book. So we have some of the equations and some of the explanation about the pH, and as you can see, we have many questions actually explaining about the energy until at the end of the day we are getting them. You see, we have few protons, we have many protons. pH is 8, pH is 7. Lower pH generates the we have concentration gradient and chemical gradient. All of these, they pull the electrons to inside, and that pull of electrons, because this membrane is impermeable, so will pass through the Stokely particles. At the end of the day will form ATP. So this marks the end about the discussion on the electron transport chain in detail. However, our discussion about the electron transport chain doesn't end here. So let we break for a little, about few minutes, then we will continue. Nataka tuendelee na mitochondria and electron transport chain. What we will go to explain is about the same as what we have talked here, lakini kwa wale ambao mnataka kwa deep, endelea kustay. Tuna break kama dakika 5, then we'll come back. Yeah, I am back. Tumalizie kwa a few minutes, 15, 20, 30. Tumalizie kuhusu oxidative phosphorylation. Now we are going to discuss about the mitochondria and the oxidative phosphorylation. Mitochondria are present in all eukaryotic cells, where they are the main sites of respiratory activity. Highly active cells require much energy, characteristically have numerous large mitochondria packed with crystal. Such cells include liver cell, Liver cell require energy to drive the large and variety of number of the biochemical reactions taking place there. Also, they included the striated muscle cell, which require energy which is needed for muscle contraction, especially where this is rapid, for example in flight muscle of insect. Also, we have the sperm tails. This provides energy to propel the sperm. We have the nerve cells. Mitochondria are especially numerous adjacent to synapse, where they provide the energy needed for the production and release of transmitter substance. Hiyo ni kwenye nerve cell. Lakini pia kuna intestinal epithelial cell ambako tunatumia energy kwa ajili ya active transport of the end product of digestive materials. So the mitochondria have the inner membrane which is enfolded to form cristae in order to increase its surface area. The cristae are lined with stocked particles. Within the inner mitochondrial membrane, they appear to be a mechanism. Kwa tunachokielezea nimekwambia akitofautiana sana kile ambacho tuna kidiscuss sio muda. So kuna mechanism ambayo energy inatengenezwa within the inner mitochondrial membrane, na ndicho kitu ambacho tunataka tukigusia hapo tukifanyia discussion. Kuna mechanism ya active transport ya protons from the matrix into the space between the inner and outer membranes of the organelle. Kama tulivyosema kule kuna zile complex ambazo kwa kadri electrons zinapokuwa zinapita tuna pump proton kwenda kwenye intermembrane space. This creates an electrochemical gradient of hydrogen ions across the inner membrane. Kwa tunatengeneza electrochemical gradient. According to the Chemiosmotic Theory put forward by British biochemist Peter Mitchell in 1961, it is the energy of this charged membrane which is used to synthesize ATP. Energy of this charged membrane, yani accumulated hydrogen in the intermembrane space, which is used in synthesis of ATP. Basically, the hydrogen atoms are picked up by NAD in the matrix and later split into protons and electrons. So the protons enter the space between inner and outer membrane of the mitochondria, while the electrons pass along the cytochrome located within the inner membrane. The protons flow back to the matrix via stocked granules, owing their high concentration in the membrane space. Kwa tunavyokuwa tunasafisha protons kutoka kwenye intermembrane space kurudisha kwenye matrix, tunatengeneza nini? ATP. Kwamba this flow act as a driving force to combine ADP and inorganic phosphate and so synthesize ATP. ATPase associated with the stocked granules catalyzes this reaction. The protons then recombine in the matrix with the electrons, and the hydrogen atoms so formed they combine with oxygen to form water. Kwa unaweza kuona hapo jinsi gani ambavyo pumping ya protons inapelekea release of energy, na hivi ndivyo ambavyo ina mitochondrial membrane inakuwa lined with the crystal, then crystals zinakuwa zina stocked particles hapo, zinakuwa zina stocked particles. Kwa hiyo nataka tu nielezee, tumalize hapa. I know you are tired about the electron transport chain, lakini tumalizie. So hivi ndivyo ambavyo process inakuwa inatokea kwenye inner mitochondrial membrane. Unaweza kuona kwamba tunakuwa tuna hydrogen atoms from Krebs cycle hapa. Hydrogen atoms from Krebs cycle zinaingia hapa. So what happens manake electron zinakuja hapa, hydrogen inakuja hapa. Kwa hiyo hii hapa unayoiona ni crystal. Hii hapo unaiona yote, hii ni crystal. Hii yote ni crystal. So unavyoona hiyo crystal, maana yake ni kwamba kwenye hii crystal yetu hapa hapa, hii ni space ambayo inaenda kuungana na nini, na intermembrane space. Hii diagram iko kwenye understanding biology. Kwa hii inaenda kuungana na intermembrane space, iko kwenye understanding biology. So crystal hapo inaongeza surface area ile pale, inaingia pale, inaenda kwa vile pale. So from there ukitoka hapo unakuja kuona electron inapita, ikimaliza inarudi. Maana hapa katikati ndio kuna electron transport chain. Hapa kuna electron transport chain, lakini proton akishakuwa pumped, akija huku, hii proton huyu yeye na yeye anapita, lakini lazima apitie pale kwenye stocked particle. Akipita tu pale, anapokuwa anatoka huku nje, kuna nini, kuna coupling. Kwa anapokuwa anapita hapo, kuna coupling. Tunatoa ADP, tunatumia energy ya coupling, tunatengeneza nani, ATP. Hivyo ndivyo ambavyo ATP inatengenezwa kwenye electron transport chain. Kwa unaona, we combine then electron, hydrogen, oxygen, we are forming water. This is the outer mitochondrial membrane. Na hivi ndivyo ambavyo process inakuwa inatokea. This is how the actual process ya electron transport chain inakuwa inatokea. So this is the synthesis of ATP according to the Chemiosmotic Theory of mixture. Chemiosmotic Theory of mixture. Now, let I say, it's enjoyable. I am announcing the end of this session. I know the session was longer, but I hope you have understood many details about the electron transport chain, and it will be easier for you to tackle any question in your exams about the electron transport chain. In the next session, we will start discussing about anaerobic respiration. We shall see what happens if metabolism of glucose happens in the absence of oxygen, if metabolism of glucose takes place in the absence of oxygen. And we discuss the concept of fermentation, types of fermentation. We have alcoholic and lactic acid fermentation, and we will have some of the difference about the glycolysis and the fermentation, and some of the similarities about the two types of fermentation and their differences. And then from there we'll move to other parts of this topic. We'll move to other parts so that we can be understood with each other. Don't forget to share this session to your fellow students, and if you need these notes, you can visit the Kipaj app. So you can go to Play Store. Let me just help you to how to get the notes. Now, how to get the notes? Yeah, let me just you help you a little. Now, how to get the notes? How to get notes? Go to Google Play Store, then after that download Kipaj app. Then you can sign up, or in other words we say create account with your phone number, create account with your phone number. Then after that, next, login. Kama ukiwa umesha login, ukiingia ndani, search for, my name is Helbert Mlelwa, so I'm just using the name Dr. Mlelwa. Search for Helbert Mlelwa and then look for the gaseous exchange. If you need the book, it is somehow huge, so it is very difficult for me to send in the Telegram group. So if you need, you can just contact me directly. I think that will be the best way. Yeah, if you need the book, contact me directly and then I can send to you. So our reference books here, we have Understanding Biology, you have BS, you have the cell biology molecular approach, and let me show you this book. Let me show you. You can see how the cover page is. I want to show you the cover page of this book, 8th edition. Now this is how the book tend to appear. This is how the book appears, and I want to decrease the size here so that you can see the whole page. Yeah. So yeah, ready to this. So this is how the book looks like. Unaweza kuona hichi ndio kitabu ambacho nakitumia. Kwa kuna BS, kuna Understanding na kuna hii the Cell a Molecular Approach ambacho nakitumia kuelezea baadhi ya mechanism kwenye kile kipande tu. Kwa hiyo kuna kipande pia chingine cha beta oxidation, ameandika vizuri beta oxidation, ameandika vizuri sana. Kwa hiyo hii ni beta oxidation ya kwenye fat. Hii mechanism yote ni beta oxidation, nayo tutatumia hichi kitabu. Kwa hiyo stay tuned. Thank you.